Now let's understand what happens when a neuron receives a stimulus. When a stimulus is applied at a particular site on the polarized membrane plates, call this site A, something very important happens. The membrane at that site undergoes rapid changes in its permeability to ions. Specifically, the membrane becomes freely permeable to sodium ions. How? Voltage-gated sodium channels in the membrane open in response to the stimulus. When these channels open, they create pores in the membrane that allow sodium ions to flow through. Before the stimulus, these channels were closed. After the stimulus, they suddenly open. Once the sodium channels open, sodium ions, which remember are in very high concentration outside the axon, suddenly have a pathway to get inside. They rush in following both the concentration gradient from high concentration outside to low concentration inside, might say, and the electrical gradient, positive charges moving toward the negatively charged inside. This influx of sodium is rapid and massive. In just a fraction of a millisecond, many sodium ions flow into the axon. What is the consequence of this massive sodium influx? The inside of the axon, which was very negative, suddenly fills with positively charged sodium ions. These positive charges neutralize the negative charges that were inside. More than that, they actually exceed the negative charges and make the inside become positive. This complete reversal of the membrane polarity is called depolarization. At the resting state, remember the inside was negative and the outside was positive. Now, after sodium influx, the inside is positive and the outside is negative. The potential has reversed. We say the membrane is depolarized because the potential difference between inside and outside has been dramatically reduced and even reversed.